Welcome back to the channel Kingdom Zoology. So today's nervous system discussion will be about neurotransmitters. In this chapter, what we've done till now is structures of neurons, along with that the mechanism of impulse generation and conduction across the neurons. In the previous lecture, I also told you about the types of neurotransmitters, which will be of two types, excitatory and inhibitory. So today's lecture would be actually going forward, taking forward the concept of neurotransmitters only, the various types of neurotransmitters, what happens to them after their release. So as we have already learned in this chapter, this is the structure of a neuron, and these are your various processes of the neurons, dendrons, the main cell body or the cyton, the axon part, and these here are the axon terminals or the synaptic envelopes or synaptic knobs. And what the synaptic envelopes contain are certain chemicals which will be membrane bound. The lipid membrane is present over these chemicals. The lipid membrane bound chemicals are called synaptic vesicles. So as a stimulus is collected on the dendrons, there is generation of action potential on the axon. That action potential travels along the length of the axon up to the presynaptic membranes, which are these membranes of the synaptic envelopes. And as a result of that, calcium ions go in. They trigger the attachment or precipitation of synaptic vesicles on the presynaptic membrane so that the neurotransmitter chemicals stored inside them will start coming out. So here is the neurotransmitter chemical which has come out of the neurons, and this is supposed to attach to the target organ. The target organ that I told you in the previous class can be a muscle, or it can be another neuron, or it can be a gland also, whose secretions are to be controlled by the neuron. So as the neurotransmitter attaches here, it is going to basically attach to the protein receptors on the target organ. These are the neurotransmitter receptors on the target organ. So when the neurotransmitter goes and attaches to these receptors, it is going to basically trigger some action in the target organ. Now what reuptake word over here means is that once a neuron has released a neurotransmitter, the neurotransmitter will go and attach to the target organ's membrane and will basically regulate the actions of the target organ. After regulation of the actions of the target organ, what exactly is going to happen to the neurotransmitter? Will it linger on here forever? Obviously not, because the target organ, let's say, is a muscle, and the neurotransmitter is actually contracting the muscle. Now, in that case, contraction alone is not the activity of the muscle. Relaxation has to follow. Imagine your jaw muscles contract when you bite or chew the food, and then they cannot relax. You will not be able to open your mouth. Similarly, imagine you will be actually contracting your biceps muscle to fold your arm towards your shoulder joint. And once the biceps muscles contract and the arm gets folded over here, if you fail to relax the muscle, you cannot open your arm back again on the elbow joint. So this tells us that contractions and relaxations of muscles have to occur side by side or alternately. So if a neurotransmitter, for example, is triggering contraction of the muscle, who's the target organ in this example we are discussing, the neurotransmitter has to be removed from here to take the muscle into relaxation. Okay, and that is where this concept of reuptake comes into picture. So once the neurotransmitter is released by the neuron, once the neurotransmitter is released by the neuron, what it does, as you can see in the picture, is it attaches to its receptors on the postsynaptic membrane. What exactly are the receptors? Mostly proteins or enzymes. So the neurotransmitter chemical attaches to its receptors on the postsynaptic membrane. And what was the potential on the target organ? Same as the resting potential of a neuron, minus 70 millivolt. So the target organs also have exactly same reasons behind existence of the resting potential as was the situation with the nerve fiber or the neuron. So this is the resting potential of the target organ. As the neurotransmitter comes and attaches over here, now there are two options. The neurotransmitter can open up sodium ion channels, triggering entry of sodium. If sodium starts coming in, the potential starts rising. So that means what is generated on the target organ membrane now? Excitatory stimulus. So this new potential that is generated on the target organ, which is basically, let's say, around plus 30 millivolt, the action potential. So what do we call the action potential as? Excitatory postsynaptic potential. Because what are the membranes of the synaptic end bulbs? Presynaptic membrane. What are the membranes of the target organs called? Postsynaptic membranes. So postsynaptic membranes have a resting potential of minus 70 millivolt. The neurotransmitter chemical has to change this potential. And there are two methods by which this change can happen. Number one, opening of sodium ion channels, triggering entry of sodium, raising the potential of the membrane of the target organ. When the potential rises, this is called excitatory postsynaptic potential. And that will basically trigger some action in the target organ. But then there are certain neurotransmitters whose receptors are going to trigger opening of potassium ion channels. If the neurotransmitter attaches to receptors and the receptors trigger opening of potassium ion channels, there will be straight away potassium exit from the resting membrane. So because of potassium exit, we can notice that the outside becomes excessively positive and the inside becomes excessively negative, and the potential may fall from minus 70 millivolt to minus 90 millivolt. This potential is called inhibitory postsynaptic potential, IPSP, because of which the target organ will not be able to do any activity at all. So such neurotransmitters that generate EPSP are called excitatory neurotransmitters, and the neurotransmitters that generate IPSP, that means drop the potential below minus 70, they are called inhibitory neurotransmitters. So that is what I told you in the previous class also, that neurotransmitters, the correct word is regulate the activity of the target organ. We should not be using the terms like activation, deactivation, stimulation, because neurotransmitters can do both the things. We had mentioned a few examples also: adrenaline, acetylcholine, the common excitatory neurotransmitters, and GABA, that is gamma aminobutyric acid, dopamine, glutamic acid, glycine. These are the common inhibitory neurotransmitters. So let's put it down into writing. All this concept that I just made you understand: once the neurotransmitter is released by the neuron, it attaches to its receptors on the postsynaptic membrane, and now it may generate an EPSP, that stands for excitatory postsynaptic potential, on the membrane of the target organ, which is also called the postsynaptic membrane. This is the postsynaptic membrane. So if we look at the word clearly or carefully, excitatory postsynaptic potential. On the postsynaptic membrane, a potential is generated that is basically excitatory in nature, so that the organ gets excited for performing or stimulated for performing a function. And EPSP is any potential above minus 70 millivolt, which means we are basically raising the potential. So once EPSP is generated, The target organ is stimulated for action. Target organ is stimulated for action. These type of neurotransmitters are called excitatory neurotransmitters. These type of neurotransmitters, as we have done earlier also, the examples, acetylcholine and adrenaline, these neurotransmitters are called excitatory neurotransmitters because effectively they are exciting or stimulating the action of the postsynaptic organs or the target organs. That is option number one, which a neurotransmitter has. And this option is going to work when the neurotransmitter attaches to attaches to receptors, and those receptors open up the sodium ion channels, raising the potential of the sarcolemma above -70 millivolt. But there is one more option that the neurotransmitters have. A neurotransmitter comes and attaches to its receptors in the postsynaptic membrane, and now it may generate a new potential that would be lower than -70 millivolt. That means dropping it down further to -75, -80, -85, -90 maybe. So neurotransmitters may generate an IPSP. IPSP will be inhibitory postsynaptic potential, and this inhibitory postsynaptic potential will be any potential of the sarcolemma that is lower than -70 millivolt. And this is basically triggered by opening up of the potassium ion channels, right? So once the neurotransmitter generates an IPSP on the target membrane or on the membrane of the target organ, we will notice that the target organ will not act at all. If there was an action already going on, that will stop, or if there was an action that is supposed to happen, that will not happen. So the target organ is prevented from acting. And such neurotransmitters are called inhibitory neurotransmitters. Because what they will do now is they are going to make it impossible or very difficult for the target organ to perform an action even if it gets an excitatory neurotransmitter. Because an excitatory neurotransmitter is capable of raising the potential from -70 to +30. But if by releasing an inhibitory neurotransmitter, you have already dropped down the potential to -90 millivolt, raising it from -90 to +30 obviously is going to be more difficult compared to what this change was because over here, how much total change do you require? 100 millivolt from -7 to +30. But now how much change or spike in the potential do we require? 120 millivolt. So generation of IPSP, that means any potential that is lower than -70 will make it difficult for the target organ to work in future also. So these are the neurotransmitters that are called inhibitory because what are they generating? IPSP. These are called excitatory because what are they generating? EPSP. So that's another way of understanding the inhibitory neurotransmitters and excitatory neurotransmitters. But once again, as I just told you a while ago, let's say we take an example of an excitatory or let's say stimulating neurotransmitter, the one that would be exciting the postsynaptic membrane of the target organ, like a neurotransmitter that will be attaching to the sarcolemma of a muscle fiber and will trigger the contraction. So the neurotransmitter in this case will be called excitatory neurotransmitter. But then as we were discussing at the beginning of the session today, if the excitatory neurotransmitter is not removed, your target organ, which is the muscle will permanently stay in contraction. So, what are we supposed to do? Digest the neurotransmitter that is present over here. But for digestion, what do you require? Enzymes. And where should those enzymes be? In this area called the synaptic cleft. So, moving on to the next concept now, the synaptic cleft. Synaptic cleft, the 10 to 20 nanometer gap between the presynaptic and the postsynaptic membranes on a chemical synapse. Synaptic cleft has enzymes which help to hydrolyze the hydrolyze means digest or break down the neurotransmitters. So synaptic cleft has enzymes which help to hydrolyze the neurotransmitters. For example, let's just quote a few here. Acetylcholine esterase. Acetylcholine is the neurotransmitter. Esterase is the enzyme that is present in the synaptic cleft to digest it. So acetylcholine esterase hydrolyzes acetylcholine. Similarly, we have one more enzyme in the synaptic cleft, monoamine oxidase. Monoamine oxidase is the enzyme in the synaptic cleft that commonly hydrolyzes adrenaline. Just a few examples. Any neurotransmitter that is released in the synaptic cleft must be digested, and the digestion is done by the enzymes like acetylcholinesterase and monoamine oxidase. Now, let's say we talk about these enzymes. Once acetylcholinesterase hydrolyzes acetylcholine, or once monoamine oxidase hydrolyzes adrenaline, the breakdown components of these neurotransmitters. Because when the neurotransmitters are digested, they will be obviously breaking into something. So the breakdown components of these neurotransmitters are absorbed. Absorbed back by the axons, synaptic and bulbs. This event is called reuptake of neurotransmitter. The breakdown components of the neurotransmitter are reabsorbed by the axons, synaptic and bulbs. That's the reuptake. And what will the axons do with them? The axons convert these components into fresh neurotransmitters again. The axons convert these components into fresh neurotransmitters again to release them next time. Plus, as a result, what happens now is the activity the activity shown by the target organ is now reversed. If the target organ was stimulated by the neurotransmitter, it gets destimulated. If the target organ was inhibited by a neurotransmitter, the inhibition ends and it's now ready to get excited again. Okay, so that's basically the different examples of enzymes that would be actually helping us to bring the target organ back to its original status, relaxed, inhibited, whatever it was. So that's it, guys. This was the various types of neurotransmitters, the enzymes in the synaptic cleft that digest them, and the process of reuptake. Now, at the end of the session, I would basically give some thought-provoking question to you today, and that is, if a person is suffering from a disorder called myasthenia gravis. Myasthenia gravis is a disease that's an autoimmune disease of the locomotion system. You must have read about it in the locomotion and movement chapter. If not, then you can always refer to my movement and locomotion notes to understand what myasthenia gravis is. Myasthenia gravis patient is given acetylcholine esterase inhibitor. Myasthenia gravis patient is given acetylcholine esterase inhibitor. That means inhibition of acetylcholinesterase enzyme is done in them. The homework that you have, which you need to basically explore, is how is this going to help the person? When you give acetylcholinesterase inhibitor enzyme to the patient who is suffering from myasthenia gravis, does the situation get reversed? And if yes, why or how? So that is food for thought. So that's it for now. We'll basically keep it short today. And the next video would be concerning various parts of the brain, central, somatic that is divided into peripheral and autonomous. So we will continue with that in our next lecture. That is all for today. Keep studying, keep sharing about the channel to your friends. And remember that every single lecture that you attend over here is taking you a step closer to your dream of getting into a medical college. All right. Thank you, everyone. All the best.